Good morning one and all, welcome to our presentation. Let me start by saying a few words about our team. My name is Shushrit and these are my friends Chakrapani, Shivani, Vaishnavi, Farah. We are from BSc Life Science second years representing the Department of Microbiology of Babu Jagjeevan Government Degree College. As you all see on the screen, our topic is biological effects of essential oils. Today we are going to present our research product within a couple of minutes. Introduction. Essential oils are volatile natural compounds produced by the aromatic plants, generally called as secondary metabolites. They are characterized by strong odor. The term oil was originated in the 16th century by Spanwan, a physician. Around 3000 essential oils are known, in which the 300 are widely used in different fields like food industries, cosmetics, pharmaceutical and agriculture. The essential oils are produced by the aromatic plants which are richly grown in the warmer countries like tropical countries and Mediterranean countries. The process which involved in producing essential oil are steam distillation, hydrodistillation methods.
Constituents of essential oils. The major components are classified as two groups of biosynthetic origins. They are terpenes, terpenoids, aromatic and aliphatic hydrocarbons. Terpenes, these are also called as hydrocarbons, which are made up of combination of isoprene units. They are monoterpenes, diterpenes and triterpenes. Hydrocarbons are made up of two basic structures called carbon and hydrogen. They are also divided into two subclasses, aliphatic and aromatic hydrocarbons. Aliphatic hydrocarbons are made up of linear chain molecules and aromatic hydrocarbons are made up of aromatic rings. Example, naphthalene. Effects of essential oils. They are antimicrobial. They are effective against gram-positive and gram-negative bacteria. Examples like clove and cinnamon oil. They are also antioxidants. Essential oils have compounds like phenolics and terpenes. They neutralize free radicals. They are anti-inflammatory. They reduce inflammation. Anti-cancer. They induce apoptosis in cells. They are also cytotoxic. They damage the cell membrane. They are applied in therapeutics, inflammation and cosmetics and healthcare.
Activity of essential oils on bacteria. The gram-negative bacteria is more resistant than the gram-positive bacteria. Now I will explain the activity of essential oils on bacterial cell wall. So here gram-positive bacteria allows the hydrophobic molecules to enter into the cell wall and act on cell and cell wall. So it leads to the cell degradation, decomposition and the cytoplasmic membrane coagulation. Whereas the gram-negative bacteria, it contains the peptidoglycan layer in its cell wall and it also contains an extra layer called oligomurin and it helps in the resistance to the essential oils. The essential oils also alter the membrane permeability and it will destroy the electron transport chain. Some potential mechanism of action of these essential oils and the components in cell targets are: they will help in the fatty acid alteration and effect on the membrane proteins and increase in the permeability, coagulation, and it is also exhibits the antiquorum sensing activity.
Methods for detection of antibacterial activity of essential oil. Agar diffusion method, it is of two types, disk diffusion assay, well diffusion assay. In disk diffusion assay, sterile filter paper is impregnated with essential oil and placed on agar, inoculated with bacteria. After incubation, inhibition zones are measured. Well diffusion assay, here wells are punched into the agar media and filled with essential oil. Inhibition zones are measured similarly. Next method is broad dilution method. It is also of two types, minimum inhibitory concentration, minimum bactericidal concentration. First one, minimum inhibitory concentration. In this, essential oils are diluted with broth with bacteria. The lowest concentration inhibiting visible growth is recorded. In minimum bactericidal concentration, samples of MIC tubes are placed on agar. The lowest concentration killing bacteria is identified. Third method is microdilution method. This method is to detect bacterial viability by using calorimetric indicators like azurin performed by 96 well plates. Last method is time killing assay. Bacteria are exposed to essential oil over time. The samples are cultured to assess bacterial reduction.
Methods for detection of antifungal activities of the essential oil. The agar-based method, classified into three methods, in which the first one is disk diffusion method, in which the fungal spores are spread on the agar plate. Disc-shaped sterile filter paper placed on the agar after the incubation, measuring the zone of emission. The second one is well diffusion method, in which a borer is used to create the well, pouring the essential oils after incubation, measuring the zone of emission. The third method is poisoned food technique, which is also called as agar dilution method, in which the essential oils are mixed molded agars at different concentration. The fungal specimens are added after incubation, measuring Identifying the lowest concentration of essential oil at which the fungal specimens cannot be grown. Broad-based method, in which the first one is broad dilution method, which is also called as minimum in vitro concentration, in which the serial dilution is performed, preparing the broth, adding the fungal spores after incubation, identifying the lowest concentration at which the fungi cannot grow. The second method is minimum fungicidal concentration test, in which the sample from MIC taken, prepared on the fresh agar after incubation, identifying the lowest concentration at which the fungi cannot grow.
Now that we have discussed about the essential oils, now let us look at into the future prospects and challenges of essential oils. Future of essential oils is bright with numerous trends and innovation shaping in the industries. Personalized aromatherapy, which is used to treat a targeted treatment for the individual, and for scientific validation, which ensures the safety and the effectiveness of the product of the essential oils. Innovative delivery methods, which can ensure the treatment of the pregnant women and can better their pregnancy and precision. Innovative integration with modern medicines. Essential oils can also integrate with modern medicines with traditions of medical practices, which can help into minimize the adverse effects of any treatment or therapy sections. Conclusion: effects are doing for further exploration for the essential oils users and for future generations, and novel approaches are ongoing on chemotherapy and chemo preventions, which can use for the cancer treatments.